For decades, billions of dollars have been invested in engineering therapeutics to clear amyloid beta plaques from the brain. Yet, these interventions consistently fail to halt neurodegeneration in the vast majority of Alzheimer's patients. Clinical outcomes have underperformed to a startling degree. In several high-profile trials, the forceful removal of amyloid plaques paradoxically worsened cognitive decline and accelerated the physical atrophy of the brain. The amyloid model also fails to resolve a major genetic anomaly, the ApoE4 allele. While it is the strongest genetic risk factor for the disease, the precise molecular mechanism linking this gene to brain decay has remained obscured for over 30 years. This impasse suggests that the prevailing model is incomplete. By focusing on the plaque, researchers may have missed the primary genetic and structural drivers of the disease. A unifying alternative framework is now emerging from the National Institute on Aging. The ApoE, ApoER2, lipid peroxidation, and signaling disruption hypothesis. This chart contrasts the two models. The traditional amyloid cascade positions amyloid beta accumulation as the primary driver of tauopathy. On the right, the new pathway identifies lipid peroxidation and receptor crosslinking as the initiating lesions. In this model, amyloid is demoted to a downstream consequence. This thesis posits that sporadic Alzheimer's is not caused by toxic amyloid production, but by a structural vulnerability in lipid transport that triggers a localized intracellular kinase collapse. To understand how this collapse begins, we must look past the plaque and examine the atomic structure of a single transport protein. In the central nervous system, ApoE is the principal lipid transporter. It is tasked with trafficking cholesterol and highly vulnerable polyunsaturated fatty acids to neurons for synaptic remodeling and membrane maintenance. Humans possess three major variants of the ApoE gene: E2, E3, and E4. E4 carriers face an exponentially higher disease risk. The difference is profound. ApoE2 and E3 possess cysteine residues, while the E4 isoform contains 0 cysteines. Cysteine allows E2 and E3 to form covalent disulfide bridges, which physically conceal and shield the vulnerable lipid cargo from oxidative attack. But ApoE4 remains a monomer, leaving its lipid cargo structurally exposed to severe peroxidation. The ApoE4 allele drives an exponential increase in neurodegenerative risk through a catastrophic loss of atomic lipid shielding capacity. When those exposed lipids under peroxidation, they degrade into toxic reactive aldehydes, like 4-HNE. These compounds act as aggressive chemical agents at the synapse. These aldehydes are released exactly where ApoE docks with its neuronal receptors, primarily ApoER2. The binding domain of this receptor is uniquely rich in lysine and histidine residues, the primary targets for aldehyde attack. The reactive aldehydes act as chemical bridges, attacking the nucleophilic targets on the receptor interface. This generates an irreversible acid-stable pyrrole crosslink, covalently fusing the ApoE ligand to the ApoER2 receptor. Because these crosslinks are stable even in low pH, the complex cannot dissociate inside the endosome. This arrests receptor recycling, effectively trapping the receptors within the cell's internal compartments. This single biochemical fusion is the true nexus of the disease, physically blinding the neuron by paralyzing its synaptic receptors. ApoER2 serves a dual function. It internalizes lipids, and it operates as the primary postsynaptic receptor for Reelin, a master glycoprotein that is indispensable for synaptic plasticity. When peroxidized ApoE crosslinks and occludes these receptors, Reelin is physically blocked from binding, entirely abrogating its survival signal. Without Reelin, DAB1 accumulates pathologically, choking off the PI3K-AKT survival pathway. This removes a critical molecular break, allowing GSK3β to become hyperactive. GSK3β then executes a dual strike, hyperphosphorylating tau to unravel microtubules, and phosphorylating PSD95 to disassemble excitatory synapses. Simultaneously, the starvation of the LIMK1 pathway leads to the collapse of the actin cytoskeleton into neurotoxic rods. The tauopathy and synaptic failure of Alzheimer's are not caused by amyloid toxicity. They are the downstream execution orders of a starved Reelin ApoER2 signaling cascade. This model challenges the popular prion-like spread hypothesis. If tau pathology spreads transsynaptically, it should move blindly across connected networks. But in the human brain, it reliably skips over millions of heavily interconnected neurons. High-dimensional mapping using multiplex immunohistochemistry allows researchers to probe dozens of these biomarkers at once within a single tissue section. Mapping reveals early tau pathology originates exactly in isolated neurons with exceptionally high ApoER2 expression. Multiplex imaging proves phosphorylated tau strictly co-accumulates with trapped DAB1 and cross-linked ApoER2 within these same cells, a direct biochemical footprint of local signaling failure. Tau does not spread like an infection. Specific neurons independently collapse because their lipid-hungry receptors have been terminally occluded. As occluded receptors and peroxidized lipids pile up outside the cell, they form the dense cores of the neuritic plaques first identified by Oscar Fischer over a century ago. Within this framework, amyloid beta must be reconsidered. At physiological concentrations, it is not a rogue pathogen, but a potent antioxidant and metal chelator. The BASE1 enzyme, which produces amyloid beta, is a stress response protein that neurons heavily upregulate in direct response to oxidative stress. As toxic lipids accumulate, neurons intentionally secrete amyloid beta monomers to bind, chelate, and neutralize the oxidizing cargo, preparing it for clearance. Amyloid beta secretion is a desperate protective response. It only transitions into toxic plaques when the brain's antioxidant capacity is overwhelmed by the unprotected lipids of ApoE4 carriers. Forcefully stripping amyloid beta from the brain without addressing the underlying lipid peroxidation effectively removes the synapse's final defense. This leaves the synapse exposed to unchecked oxidative destruction. The new therapeutic pipeline must prioritize stabilizing the Reelin ApoER2-Dab1 axis and mitigating lipid peroxidation before crosslinking can occur. This means developing targeted ApoER2 agonists, using lipid scavengers to quench aldehydes, and employing gene editing to restore disulfide bridges. Addressing the root atomic vulnerability in lipid transport offers the first biologically sound roadmap to restoring synaptic homeostasis. By abandoning the linear assumptions of the amyloid cascade, the neuroscientific community can finally breach the theoretical stagnation and arrest Alzheimer's disease at its true origin.